Hi students, myself Chandrasekhar, Zoology faculty from Sri Chaitanya. In Sri Chaitanya's NEET Sankalp 2.0 program, today we are moving forward with a new chapter, excretory products and their elimination. Let's get into the topic. Animals accumulate the nitrogenous waste like ammonia, urea, uric acid, creatinine, and they also accumulate carbon dioxide, excess water, ions like sodium, potassium, chloride, and some other salts like sulfates, bicarbonates. And these components, they have to be eliminated from the body either partially or totally. And different organisms have different mechanisms for this elimination. And this elimination of nitrogenous waste and all these excess components from the body is known as excretion. So in this chapter, the first topic I am going to discuss is the organisms, type of organisms based on the excretory product produced in them. Organisms are classified into three categories broadly based on the type of excretory product: ammonotelic organisms, ureotelic organisms, uricotelic organisms. The process of excreting ammonia is called ammonotelism. The example organisms that are excreting ammonia includes many bony fishes, aquatic amphibians, aquatic insects. They are ammonotelic in nature, and early invertebrates like sponges, cnidarians also excrete ammonia. And ammonia can be excreted in the form of ammonia and also in the form of ammonium ions. And ammonia is readily soluble in water. It is highly toxic in nature, and for the elimination of ammonia, the animals require high amount of water. That is the reason why ammonia is the major excretory product of aquatic organisms. In fishes, kidneys do not play significant role in the elimination of ammonia, because ammonia can be eliminated through the gill surface in the form of ammonium. So these are the points regarding ammonotelic organisms. When it comes to the ureotelic organisms, animals mainly excreting urea are called ureotelic organisms. See, the examples of ureotelic organisms includes mammals, including humans. We are ureotelic in nature. Many terrestrial amphibians, example frogs, marine fishes like Pristis, Scoliodon, etc., are ureotelic in nature. They are ureotelic in nature. See, urea is moderately soluble in water and it is moderately toxic. That means it is less toxic compared to the ammonia. And for the elimination of urea, moderate amount of water is required. The urea is synthesized in our body by a particular process called ornithine cycle. It occurs in our liver. Ornithine is a kind of non-protein forming amino acid that is a main component in this cycle, hence it is called ornithine cycle. So the urea composition is NH2 CO NH2. So two amino groups and a carbon dioxide is required for its elimination. That means the liver takes ammonia from amino acids of blood, converts it into urea, releases the urea back into the blood. From blood, urea goes to the kidney and then eliminated. And the synthesis of urea in the liver is called the process called ornithine cycle. Urea is synthesized by the liver, urea is eliminated by the kidney. Next, the third one is uricotelic organisms. Animals excreting uric acid are called uricotelic organisms. The examples includes reptiles, birds among the vertebrates. Among the invertebrates, land snails and terrestrial insects. They are uricotelic in nature. Among the terrestrial insects, in our syllabus we have cockroach. Cockroach is uricotelic in nature. It is less toxic, less soluble in water, and least amount of water. Even without water, the uric acid can be sent out. See, cockroach can send uric acid in the form of pellets. So without water it can be sent out. That means the uric acid excretion is involved in the water conservation. So that excretion of uric acid is said to be the best terrestrial adaptation for the conservation of water. The next one is excretory organs in different organisms. Protonephridia are the excretory organs. The protonephridia are the simple tubular structures, and they are having a structure called flame bulb, also known as the flame cells. They are primarily involved in osmoregulation, and the next function of this protonephridia is excretion. Organisms showing protonephridia includes planarians, that means flatworms, they belongs to the phylum Platyhelminthes, rotifers, some annelids, it is not earthworm, because earthworm near is leech, they are having nephridia, they are not having protonephridia, and some annelids, and lancelets. Lancelets belongs to the subphylum Cephalochordata. The example for that is Amphioxus. They are having protonephridia, simple tubular structures, as their excretory organs. The next one is nephridia. The nephridia are mainly present in earthworms and other annelids like Nereis and leech. Both are having nephridia. Nephridia also involved in both excretion and osmoregulation. Next, Malpighian tubules. Malpighian tubules, they are the tubular structures, fine thread-like structures present in insects. Most insects, including cockroach, they perform excretion with the help of Malpighian tubules, and Malpighian tubules are also involved in both osmoregulation and excretion. Next one is antennal glands, also known as green glands. They are present in aquatic arthropods like crustaceans, like prawns and crabs. At the base of antenna, there is a gland which is responsible for the excretion, known as antennary glands. When it comes to the vertebrates, you all know that the excretory organs are kidneys, cyclostomes, fishes, amphibians, reptiles, aves, mammals, they are all having kidneys to perform the excretion. Of course, the kidneys are involved in osmoregulation also. Now let us get into the human excretory system topic. In the human excretory system, the important parts includes a pair of kidneys, a pair of ureters, a urinary bladder and urethra. These are the important parts. Let us talk about kidneys first. A pair of kidneys present in the abdomen of human beings on either sides of the vertebral column from last thoracic vertebra to the third lumbar vertebra. See, if it is one kidney, this is another kidney, last thoracic, first lumbar, second lumbar, third lumbar. Kidneys are extended on either sides. Kidneys are called retroperitoneal organs because they are covered by the peritoneum on their ventral side. They are bean shaped, reddish brown in color. Measurements of kidneys, length of the kidney is around 10 to 12 centimeters. Next, width of the kidney is 5 to 7 centimeters and thickness is 2 to 3 centimeters. On an average, each kidney consists of 170 grams weight. So, this is regarding kidney location. Next, if you see the external, internal and microscopic structure of the kidney, kidneys are covered by a renal capsule for protection. The outer side of the kidney is convex and inner side of the kidney is concave. On the concave side of the kidney, there is a notch present known as hilum. Blood vessels, ureter, nerves enter the kidney through this hilum, and kidney is protected by renal capsule. And inside the kidney is having two portions, outer cortex and inner medulla. The medulla is in the form of a conical processes. See, this is the ureter. Ureter inside the kidney forms a funnel shaped structure. This funnel shaped structure is known as renal pelvis. This funnel is forming cup like structures, this red color portions. These cup like portions are said to be renal calyces. There are many renal calyces present. Extending into each renal calyx, the conical masses are there which are triangular in shape. They are said to be medullary pyramids. This is one medullary pyramid. This is another medullary pyramid, and this one is cortex. The cortex is extended between renal pyramids as columns. They are called renal columns and also known as columns of Bertini. Columns of Bertini. So, this is regarding the internal structure of the kidney. Cortex, medulla, medulla extends into the Cup-like structures called calyces. Calyces are formed from the pelvis, which is a funnel. Pelvis is the broad structure which is formed by the ureter inside the kidney. And see this red color is renal artery, which is supplying blood to the kidney, and renal vein carrying the blood out of the kidney. And I will discuss the blood flow inside the kidney later. After the external and internal structure of the kidney, let us talk about microscopic structure of the kidney. In the microscopic structure of the kidney, each kidney consists of 1 million nephrons approximately. Nephrons are also called uriniferous tubules. Uriniferous tubules. And we can divide the nephron into two portions. One is glomerulus, second one is renal tubules. Glomerulus and renal tubules. See this, an arteriole extending into the kidney is called afferent arteriole. This afferent arteriole divides into a network of capillaries. That network of capillaries put together known as glomerulus. And these glomerular capillaries unite to form, and that comes out of the Bowman's capsule, known as efferent arteriole. Compared to the afferent arteriole is narrow, and glomerulus is a capillary network formed by the afferent arteriole. Next one is renal tubules. The first part of the renal tubule is Bowman's capsule. Bowman's capsule is a bowl-like structure. It is a double-walled. Bowman's capsule is lined by the simple squamous epithelium, and it is present, see this, if it is the kidney, this is cortex of the kidney, this is medulla of the kidney. The Bowman's capsule is present in the cortex region of the kidney. And the inner wall of the kidney is lined by the Bowman's capsule lined by the simple squamous cells. See, if it is the Bowman's capsule, these are called podocytes, the simple squamous cells, and between these podocytes there are minute pores present known as filtration slits or slit pores. In the wall of glomerular capillaries also there are pores called fenestrae. See this is the wall of Bowman's capsule, and this is the wall of glomerulus. This glomerular wall plus Bowman's capsule wall plus a basement membrane together forming a filtration membrane. There are three components in the filtration membrane. One is a simple endothelium of glomerular capillary, followed by the basement membrane, followed by the simple squamous epithelium of Bowman's capsule. Next, the Bowman's capsule, see, it is an extension of PCT, proximal convoluted tubule. It opens into a next part called PCT, proximal convoluted tubule. So, this is PCT. PCT is highly coiled. PCT is lined by the simple cuboidal epithelium with brush border. Because of that reason, the absorption rate is more in PCT compared to the other parts of nephron, and it is also located in the cortex region of the kidney. It is highly coiled in nature. Next, PCT opens into a structure called a loop of Henle. In the loop of Henle, there are two portions like a descending limb of Henle and ascending limb of Henle. The descending limb of Henle and ascending limb both are there in the medullary region of the kidney. Descending limb is lined by the simple squamous epithelium. When it comes to the ascending limb, in the ascending limb there is a thin part and thick part. Thin part of the ascending limb is lined by simple squamous epithelium, thick part is lined by the simple cuboidal epithelium, and it will extend as distal convoluted tubule. DCT is also coiled. DCT is also lined by the simple cuboidal epithelium, no doubt at all. And there are some specialized cells are there like principal cells and intercalated cells in the DCT. And DCT is located in the cortex region. Cortex region. See at the tip of the DCT, many DCTs opens into collecting duct. See, this is the collecting duct. A collecting duct is there in the cortex and also extends into the medulla. But collecting duct is not a part of single nephron. DCT opens into collecting tubules opens into collecting duct. So, many DCTs of many nephrons opens into one collecting duct. So, this is regarding nephron structure. See, these collecting ducts they extends into renal pyramid. See this, these black color lines are nothing but collecting ducts. They opens into the renal pyramid. In the renal pyramid, collecting ducts join to form a duct of Bellini. The ducta bellini opens into the tip of the renal pyramid known as renal papilla. This renal papilla opens into the calyx. Calyx opens into pelvis. Pelvis opens into ureter. This is the passage of urine. So, ducta bellini is formed by the joining of collecting ducts in the renal pyramid. One more part regarding the nephron is, listen carefully, at the junction of DCT, distal convoluted tubule, and afferent arteriole, the cells are crowded. This particular region is called juxtaglomerular apparatus. This juxtaglomerular apparatus is formed by the cellular modifications of distal convoluted tubule and afferent arteriole. So, in this crowded cells, listen, the cells that are crowded that are related to the DCT are called macula densa, and the cells that are crowded related to the afferent arteriole are called juxtaglomerular cells. The macula densa plus juxtaglomerular cells put together known as juxtaglomerular apparatus. And this juxtaglomerular apparatus is the inbuilt mechanism in the kidney for the regulation of glomerular filtration rate. The filtration that is happening in the at the glomerulus will be regulated by this particular apparatus. And in this, the JG cells are involved in the production of an enzyme called renin. They are involved in the production of a hormone, peptide hormone called erythropoietin. And you all know that the function of erythropoietin, it stimulates the production of RBC. And when it comes to the nephron, again there are two types of nephrons in the human kidney. One is cortical nephrons, second one is juxtamedullary nephrons. Let me give you the differences. Cortical nephrons are more numerous. Nearly 80 to 80% of the nephrons in our kidney are cortical. Remaining percentage of nephrons in our kidney are juxtamedullary. And what are the main differences? The main difference with respect to the loop of Henle and vasa recta, I just want to tell you. The loop of Henle is short in cortical nephrons. Loop of Henle is long and extends deep into the medulla in juxtamedullary nephrons. This radicular tube around the loop of Henle is called vasa recta. Vasa recta, it is either short or absent in cortical nephrons. Vasa recta is well developed in juxtamedullary nephrons. When we talk about the glomerulus and Bowman's capsule location, you know, glomerulus and Bowman's capsule put together known as Malpighian body, which is also called a renal corpuscle. That renal corpuscle, it is located slightly upper region of the cortex in cortical nephrons. The glomerulus and Bowman's capsule is located at the junction of cortex and medulla in juxtamedullary nephrons. So, these are the differences in cortical and juxtamedullary nephrons. So, with this I have completed the structure of a kidney. And after the kidney, next one is a ureters. Ureters are made up of a transitional epithelium and smooth muscles, and they conduct the passage of urine. A pair of ureters are there, and they will open into the urinary bladder. Urinary bladder is a sac-like structure located ventral to the rectum in human, and the function of urinary bladder is it temporarily stores the urine. The wall of urinary bladder is having transitional epithelium, and wall of urinary bladder consists of smooth muscles. Of course, on the wall of urinary bladder, there are some receptors which can indicate the urine accumulation in the bladder, that they are called stretch receptors. And bladder opens into urethra. See, at the junction of bladder and urethra, there is an internal urethral aperture guarded by internal urethral sphincter. At the tip of the urethra, there is an external urethral meatus guarded by external urethral sphincter, that is made up of voluntary muscles. Internal urethral sphincter is made up of involuntary muscles. The length of the urethra, it is short in females and it is long in males. Urethra opens out in females through urethral orifice. Urethra passes through the penis in males and opens at the tip of the penis through external urethral meatus. And urethra in females conducts the passage of urine only, but the urethra in males, it conducts the passage of urine. As it is connected to the ejaculatory duct, it also conducts the passage of semen. Hence, it is said to be urinogenital canal in male humans. With this, excretory system topic completed. Now, let us talk about urine formation. In the urine formation, there are three important steps. Step 1 is glomerular filtration, next is tubular reabsorption, and the next selective reabsorption, and the next one is tubular secretion. See this movement of contents from the blood of a glomerulus passing through the filtration membrane into the Bowman's capsule is called glomerular filtration. Next, after glomerular filtration, as it is random, many contents get into the renal tubes, because almost all the constituents of plasma get filtered except large plasma proteins. That is a common statement. But from that, there are many useful components may be there in the filtrate. They will be taken back into the blood by a step called selective reabsorption. But even after filtration, the blood may have some harmful components which has to be given to the renal tubules for elimination. That giving back of harmful components from blood to the renal tubules is called tubular secretion. Glomerular filtration is random, it is non-selective, it occurs by pressure gradient. Tubular secretion is generally occurs by active transport. Selective reabsorption takes place. Reabsorption means from tube to the blood. It may occur by active transport, it may also occur by passive transport. Let us get into the topic. The first one is glomerular filtration. Listen carefully. 1100 to 1200 ml of blood filtered by the kidneys per minute in humans. While that blood is passing through the glomerulus, the glomerular hydrostatic blood pressure is equal to 60 mm of Hg. 60 mm of Hg is the glomerular hydrostatic blood pressure. Actually, glomerular hydrostatic blood pressure helps in filtration, but it is opposed by two pressures. One is colloidal, see this is glomerular hydrostatic blood pressure, which is opposed by colloidal osmotic blood pressure, that is around 32 mm of Hg. It opposes, opposes means acts in opposite direction, opposes the filtration, plus capsular hydrostatic blood pressure, hydrostatic pressure, that is 18 mm of Hg. 32 plus 18 is equal to 50. 50 mm of Hg pressure is acting opposite to the 60. 60 wants to cause the filtration, 50 opposing it. That means 60 dominates the 50 by factor 10. That 10 mm of Hg is said to be net filtration pressure. Because of net filtration pressure, almost all components of plasma except plasma proteins finally filtered through this filtration membrane. Hence, this process is called ultrafiltration. And how much filtrate is formed from this 1100 to 1200 ml of blood? It is around 125 ml. This 125 ml is said to be glomerular filtrate, and it is glomerular filtration rate per minute. Per minute is 125 ml. So, what is there in this filtrate? In this 125 ml filtrate, there is water, there are nutrients like glucose, amino acids, fatty acids. Next, nitrogenous waste, many salts can be there. So, that means these nutrients, many salts and water, they are all useful to the body. So, this filtration is a random filtration, non-selective filtration, only based on the pressure gradient. And glomerular filtration rate per minute is 125 ml and per hour is around 7.5 liters, per day is around 180 liters. So, 180 liters is the glomerular filtration rate per day, but entire filtrate we will not excrete it. 99% of this filtrate which is produced in one day can be reabsorbed. 1% of this filtrate, even less than 1% can be eliminated in the form of 1 to 1.5 liter of urine. So, that means from the filtrate so many components should be reabsorbed, and some components which are not filtered, which are there in the blood, should be eliminated. For that, we have to get into the next topic. That next topic is selective reabsorption of components through the renal tubules and selective secretion of components into the tubules. Let us talk about functions of tubules, both reabsorption and secretion. When the filtrate from Bowman's capsule, when it enter, and I forgot to tell you guys, 1100 to 1200 ml of blood is filtered per minute. How much amount of blood pumped by each ventricle in 1 minute to the body parts, it is approximately 5 liters. Out of 5 liters of blood pumped by the heart in 1 minute, 1100 to 1200, approximately 1 liter is coming to the kidney, so that renal blood flow is said to be 1/5 of cardiac output. 1/5 of cardiac output. From the renal blood flow, how much filtrate is formed in 1 minute? 125 ml. 125 ml is the GFR, 1100 to 1200 ml is the renal blood flow. That is 1/5 of cardiac output. Okay, let us talk about absorptions and secretions at the renal tubules. While the filtrate is passing through the PCT, as the PCT is lined by the simple cuboidal epithelium with a brush border microvilli, it increases the area of absorption. Listen carefully. 70 to 80% of water and electrolytes are absorbed while the filtrate is passing through the PCT. And water absorption by passive transport like diffusion, sodium absorption by active transport, potassium absorption by active transport, chlorides, bicarbonates absorption can occur by passive transport. And almost all the nutrients are absorbed at the DCT, nearly 100%, like glucose, amino acids. That is the reason why the urine in a healthy person's condition should not have glucose. So, the urine will not have glucose in a healthy person because almost 100% of the glucose is absorbed at the PCT itself. And absorption of glucose and amino acids is also occur by active transport. And what are secreted into the filtrate for elimination from this cortical region? They are hydrogen ions and ammonia. They are actively secreted. And the secretion of hydrogen mainly helps in the maintenance of acid base balance of the blood, because whenever more H plus is there in the blood, it reduces the blood pH and causes acidity. Next, the PCT, from the PCT the filtrate gets into the loop of Henle and into the first part of the loop of Henle, that is descending limb of Henle. The descending limb of Henle, wall of descending limb of Henle is impermeable to water and permeable to water and almost impermeable to salts. It is permeable to water, so that the water is reabsorbed from the filtrate of a descending limb while it is moving down towards the medulla from cortex. But generally salt absorption or secretion will not occur here because it is almost impermeable. Next, when the filtrate gets into the ascending limb, the wall of ascending limb is impermeable to water, that is important, but permeable to salts. In the thin part, the sodium chloride is reabsorbed by passive transport. In the thick part, sodium chloride, even potassium ions are reabsorbed, the sodium potassium is absorbed by active transport. Reabsorption. But slight amount of urea is secreted from the inner medulla into the filtrate of ascending limb, that is secretion. Okay, fine. After this, the filtrate gets into the descending limb. Once the filtrate gets into the descending limb, from here onwards the absorption will be conditional. So that means whatever the absorption occurred in the PCT, that is unconditional and which is a mandatory absorption. Without that absorption, our organism cannot survive properly. That is the reason why the absorption that is happening in the PCT is called obligatory reabsorption. And whatever the reabsorption that is happening in the DCT and collecting duct is called facultative reabsorption. That depends on the body condition. Even after this much reabsorption, still if our body requires water and salts from this filtrate, now the absorptions can occur in the DCT and collecting duct with the help of certain hormones. In the DCT, absorption of water and salts followed by salts occur with the help of a hormone called ADH, antidiuretic hormone, secreted by the hypothalamus. Salts and also water reabsorption. See, water salts is ADH, you can take salts followed by water, you can take a hormone called aldosterone, like sodium, like bicarbonates. And aldosterone not only causes the reabsorption of sodium, it causes the secretion of potassium. That means when blood is having excess potassium, aldosterone causes its elimination. When blood is having less sodium, aldosterone causes the reabsorption of sodium from the filtrate. So sodium, chloride, bicarbonates, water absorption occur at the DCT, but this is facultative. And what are the components that are secreted at the DCT? K plus, H plus and also ammonia, they did not mention in the picture. So by this you can say DCT is involved in the maintenance of sodium potassium balance because it is causing absorption of sodium, secretion of potassium. and it is also maintains the acid base balance because it is involved in the secretion of hydrogen ions. Now the filtrate gets into the collecting duct. While the filtrate is passing through the collecting duct causes the reabsorption of water, no doubt, reabsorption of water, and also reabsorption of slight amount of urea. See, after collecting duct, they will get into the ducta bellini, finally into the renal pelvis, ureter, and sent out. But before the urine getting into those parts, from this urine, some amount of urea is reabsorbed from the collecting duct into the medullary interstitium for the maintenance of concentration gradient of medullary interstitium. And this absorption of urea is a nitrogenous waste. Nitrogenous waste absorption generally occur by passive transport. And along with that, some components are secreted into the collecting duct. What are they? K plus, H plus will be secreted into the collecting duct. So components that are secreted into the collecting duct are K plus and H plus. Components that are absorbed from the collecting duct are water and slight amount of urea. So I can say a huge amount of water is absorbed from the collecting duct, no doubt at all. But highest amount of water that is absorbed in which part of the nephron? The answer is proximal convoluted tubule. In which part of the nephron there is least absorption? The answer is ascending limb of Henle, because it is impermeable to water. And what is the main function of loop of Henle? Loop of Henle is mainly involved in the maintenance of concentration gradient. So with these functions of tubules are completed, the next topic is mechanism of concentration of urine by a process called countercurrent mechanism. See, why is it called countercurrent mechanism? See this is the loop of Henle. In the descending limb, the flow of filtrate is downward, in the ascending limb upward, forming a countercurrent. This is vasa recta. Before getting into the vasa recta, let me tell you the blood flow inside the kidney. Blood flow inside the kidney. See this. There is a blood vessel called aorta. Aorta gives the blood to renal arteries. Renal arteries. Renal artery enter the kidney and become afferent arteriole. From afferent arteriole, blood goes to glomerulus. From glomerulus, blood goes to efferent arteriole. But this efferent arteriole, see this, it is divided into capillaries. The capillary network present around PCT and DCT is called peritubular network, and the capillary network that is present around the loop of Henle is called vasa recta. So that means the blood from efferent arteriole is going to the peritubular network and vasa recta. See, efferent arteriole, peritubular network and vasa recta. From vasa recta, blood goes to renal venules. Renal venules unite to form renal vein. Renal vein opens into the inferior vena cava, and inferior vena cava to heart, again from heart to renal artery. See the blood flow to and from the kidney. First heart, heart to aorta, aorta to renal artery to afferent arteriole, then glomerulus, efferent arteriole, peritubular network. In that, vasa recta is present around the loop of Henle, then renal venules, renal vein, inferior vena cava, and then back to the heart. This is the blood flow. So regarding the vasa recta, see these guys, this is vasa recta. The flow of blood in the vasa recta in this limb is downward and here is upward, again forming the countercurrent. This portion of vasa recta is known as descending limb of vasa recta. This is called ascending limb of vasa recta. And what about loop of Henle story? This is called descending limb of Henle. This is called ascending limb, descending limb, and this one is ascending limb of Henle. Let me explain the process now. The loop of Henle is in the medulla region. PCT Bowman's capsule is in the cortex region. The cortex is having cortical interstitial fluid, medulla is having medulla interstitial fluid. When we see the concentration gradients, The cortical fluid concentration gradient is 300 milliosmoles. The filtrate which is passing through the Bowman's capsule to the PCT, it is also having 300 milliosmoles concentration. While the filtrate is moving down in the descending limb of Henle, as it is permeable to water, what is happening to the concentration of the filtrate? It is increasing because water is reabsorbed. It will become 400, 600, 800, 1000 and finally become 1200. At the hairpin limb loop of Henle, the filtrate is having highest concentration, that is around 1200 milliosmoles. While the filtrate is moving up, as it is permeable to salts like sodium chloride, as sodium chloride is absorbed along with the potassium, the concentration gradient of the filtrate will be reduced. So, 1200 to 900, 600, 400, 300. And finally, the filtrate that is about to enter into the DCT will have 200 milliosmoles. So, it is okay, fine. So, why is it and how is it making the concentration gradient? Listen carefully now. This is vasa recta, actually it is present around loop of Henle. The sodium chloride which is absorbed from the ascending limb of vasa recta will be taken by the descending limb of vasa recta. Ascending limb of Henle, it causes the reabsorption of sodium which is taken by the descending limb of vasa recta. And after taking the sodium chloride, see this, the vasa recta giving back the sodium chloride into the medullary interstitium from its ascending limb. So, that means sodium chloride is finally coming back to the interstitium, especially to the inner medullary interstitium. Along with that, you know, from collecting duct, little amount of urea is also reabsorbed. So, this sodium chloride absorbed, getting into the interstitium from this ascending limb of vasa recta, urea that is absorbed from the collecting duct, both are increasing the concentration gradient of inner medullary interstitium. Actually, in the cortex, the medullary concentration gradient is 300 milliosmoles. Slowly it will become 600, then 900 and finally it will become 1200. So, the first the function of this countercurrent mechanism is they have to increase the concentration of inner medullary interstitium finally to 1200. Initially in the cortex it is 300 to become 1200. And who is responsible for the maintenance of that concentration gradient? The sodium chloride and urea. Okay, fine. So, what happens because of increasing this concentration gradient? Now, listen carefully. The filtrate that is getting into the collecting duct is having the less concentration gradient compared to the medullary interstitium. That is the reason why, see this, the urine concentration at the tip of the collecting duct should be equal to 1200. 200 to become 1200. What should occur while the filtrate is passing through the collecting duct? Water should be reabsorbed. So, large amount of water is reabsorbed while the filtrate is passing through the collecting duct, because the concentration of the filtrate should increase and should be equal to the concentration of medullary interstitium. As medullary interstitium is 1200, to increase the concentration of the filtrate to 1200, huge amount of water is reabsorbed. So, that is causing the reabsorption of water and that is causing less water loss through the urine. That is one point. And urine concentration is increasing, hence our urine is said to be hypertonic. It is isotonic to the medullary interstitium 1200 and 1200 and it is hypertonic to the blood. So, humans produce highly concentrated urine and our urine will be concentrated 4 times when compared to the concentration of the initial filtrate. Initial filtrate concentration is 300 milliosmoles and finally the urine concentration is 1200. It is concentrated 4 times. So, this is regarding countercurrent mechanism. The first function of the countercurrent mechanism is to increase the concentration of medullary interstitium by retaining sodium chloride and urea in the medullary interstitium. That increased concentration causing the reabsorption of a huge amount of water from the filtrate while it is passing through the collecting duct, thereby increasing the concentration of filtrate 4 times to the initial filtrate. In this, the loop of Henle acts as countercurrent multiplier and vasa recta acts as countercurrent exchanger. So, this is regarding countercurrent mechanism. Now, let us talk about regulation of kidney function. Kidney function is regulated by the hypothalamus, hormones of hypothalamus, juxtaglomerular apparatus and to some extent by the heart. Let us talk about regulation without making late. First, regarding ADH. When there is loss of fluid from our body, that increases the ionic concentration, but water loss will be there from the body. When there is loss of fluid from our body, the osmoreceptors of hypothalamus get activate. That causes the production and release of a hormone called ADH. So from hypothalamus, neurohypophysis, from there it will be released into the blood. So the ADH through the blood, finally it goes to distal part of the renal tubule, that is DCT. So the DCT is having a filtrate and this one is blood. This one is blood. So what is this ADH can do in the DCT after getting into the DCT? It causes reabsorption of water into the blood followed by reabsorption of salts. When water or salts from the filtrate of DCT gets into the blood, that increases blood volume, that increases the GFR. Increases the blood volume and increases the GFR. So ADH increases GFR, increases the blood volume is correct. Another mechanism of ADH is, ADH can also cause the constriction of blood vessels. When blood vessels are constricted, that increases the blood pressure and increases the glomerular blood flow and finally increases the GFR. The overall regarding the functions of ADH, it increases the blood volume, it increases the GFR, it increases the blood pressure, So this is regarding the ADH mechanism. So as ADH is causing reabsorption of water, then there will be less water loss through the urine. When there is more loss of water there through the urine, that is called diuresis. As ADH is preventing the diuresis, it has got the name called antidiuretic hormone. Whenever there is deficiency of ADH, that causes the excess urination. Excess urination is a characteristic symptom of a disease known as diabetes insipidus. The next mechanism is renin-angiotensin system mechanism. So whenever there is decrease in glomerular blood flow or there is decrease in glomerular blood pressure or GFR, I already told you glomerular filtration rate is regulated by the JGA. The juxtaglomerular cells of juxtaglomerular apparatus produces an enzyme called renin. This renin converts angiotensinogen, which is present in the blood, into angiotensin 1 and finally converts it into angiotensin 2. This angiotensin 2 acts on the adrenal cortex. Adrenal cortex. See this. Adrenal cortex. When it acts on the adrenal cortex produces mineralocorticoids. The principal mineralocorticoid is aldosterone. Aldosterone will also go to DCT and this is blood and it causes absorption of salts like sodium and bicarbonates and absorption of water. Of course, it causes the secretion of potassium, that is a different story. But because of absorption of sodium, bicarbonates and water, it increases the blood volume and increases the GFR and it is also increases the blood pressure. One more mechanism regarding the increase in blood pressure is angiotensin 2 is the strongest vasoconstrictor. It constricts the blood vessels and increases the pressure. Overall in renin-angiotensin mechanism, there is increase in GFR, increase in renal blood flow, increase in blood amount of blood because of entry of water into the blood and also increase in blood pressure. The next mechanism is related to the hormone called atrial natriuretic factor, which is also called atrial natriuretic peptide. It is a peptide hormone. When there is a increase in blood flow to the atria, that means when there is more blood pressure, these are the atria of the heart. These atria consist of atrial myocytes. Those myocytes produce a hormone called ANF. So ANF is produced by the atrial myocytes into the blood of atria. Then the blood goes to ventricles. From ventricles, blood will be pumped to the blood vessels. ANF causes the dilation of the blood vessels. When blood vessels are dilated, that decreases the blood pressure. That is one function of ANF. But ANF, one more function is, ANF decreases the reabsorption of sodium at the DCT. As it is decreasing sodium reabsorption from the DCT, so what happens? The sodium excretion, it will be increased in the urine. Hence, it is named as natriuretic. Natri is related to sodium. So this is a ANF mechanism, but ANF mechanism is the counter mechanism to the ROS mechanism and ADH mechanism. Next, micturition. Passage of urine is called micturition. Urine formed by the nephrons is ultimately carried to the urinary bladder, where it is stored till a voluntary signal is given by the central nervous system. See, assume it is the bladder and this is the urethra. When the urine accumulates in the bladder, I already told you there are stretch receptors on the bladder. The urine bladder get expand. Now the stretch receptors will send that information to the CNS through afferent nerves, that means sensory nerves. Now the CNS gives a motor signal, that means through efferent nerves. This is a sensory signal going towards the CNS. Motor signal is the response is coming through the efferent nerves, which are also called motor nerves, towards the muscles of bladder, sphincters and urethra. So because of this motor signal, urinary bladder muscles will contract, internal urethral sphincter will get relaxed, urethra muscles contract, external urethral sphincter relaxes and opens it, causing the passage of urine called micturition. See this, the process of release of urine is called micturition, and the neural mechanisms causing it is known as micturition reflex. The urine output in a healthy person per day is 1 to 1.5 liters. The pH of the urine is 6, slightly acidic in nature, pale yellow in color, and it is having a characteristic odor. On an average, 25 to 30 grams of urea is excreted per day. And the clinical analysis of urine will give the information regarding the functioning of the kidney, regarding some metabolic disorders. One example for that is glucosuria and ketonuria. Presence of glucose in the urine, presence of ketone bodies in the urine is the indication of a hormonal disorder called diabetes mellitus, which is caused due to the deficiency of a hormone known as insulin. Role of other organs in excretion. Lungs are involved in excretion of carbon dioxide and water vapor. How much carbon dioxide will be sent out per minute? So in 1 minute, 5 liters of blood will be pumped by the ventricle, right ventricle to the lungs. Each 100 ml of blood can deliver 4 ml of CO2. 100 ml delivers 4 ml. But in 1 minute, how much blood is going? 5 liters. 5 liters means 50 hundreds. Each 100 ml, 4 ml. 50 into 4, 200 ml of CO2 will be sent out per minute in a healthy person under normal conditions. Along with that, some water vapor also will be sent out. Liver is also involved in excretion. You know, liver produces bile juice. The bile juice is stored in the gallbladder and finally released into the small intestine. It has bile salts. Salts are involved in the emulsification of fats. That's a different story. But along with that, there are some components should be excreted from the alimentary canal through the anus and out. Those components that are present in the bile juice that are excreted by the liver are bile pigments like bilirubin and biliverdin, cholesterol, degraded steroid hormones. You know, steroid hormones, once their working is over, they will be broken down in the liver. Excess vitamins and drugs, they are all eliminated by the liver through bile juice. Next, our skin consists of sweat glands and sebaceous glands. They are also involved in excretion. So the sweat glands' primary function is thermoregulation, cooling effect. But other function is through the sweat, sodium chloride, small amounts of urea and lactic acid can be eliminated. Sebaceous glands are involved in the secretion of oil. Through that oil, what are the components that are excreted through the skin to outside? Some sterols, hydrocarbons and waxes. Even our salivary glands involved in the excretion of some small amounts of nitrogenous waste like urea. So, role of other organs in excretion like lungs, liver, sweat glands, sebaceous glands and salivary glands. The next topic is the disorders of the excretory system. Regarding the disorders of excretory system, the first disorder I am going to discuss is uremia. Malfunctioning of kidneys can lead to accumulation of urea in the blood. That is called uremia, which is highly harmful and it may lead to renal failure. So, accumulation of urea due to malfunctioning is called uremia and uremia leads to kidney failure. So, what is the remedy? The immediate remedy for the uremia is hemodialysis, kind of artificial kidney. So, what is hemodialysis? Let me explain here. In hemodialysis, see, by puncturing an artery, they will take the blood into a tube called cellophane tube, which is a porous membrane tube. And this tube allowed to pass through a big tube that consists of a fluid called dialyzing fluid. So, through the cellophane tube, see this is cellophane tube, blood is passing. Around the cellophane tube, this is a dialyzer, consists of dialyzing fluid. This green is dialyzing fluid, red is blood. The dialyzing fluid is almost equal to blood plasma except nitrogenous base, and the cellophane tube is a porous membrane. See the tube finally comes out of the dialyzing unit and it will be connected to a vein. So, this is the equipment arrangement of parts like cellophane tube, dialyzing unit, again coming cellophane tube is coming and connected to the vein from artery initially. And how the process can be performed. By puncturing the artery, they will take the blood into the cellophane tube, and before draining the blood into the cellophane tube, they will add heparin to the blood to prevent the clotting of the blood outside the body. While the blood is passing through the cellophane tube, as it is a porous membrane, it allows the diffusion. Because nitrogenous bases are present in the blood, they are absent in the dialyzing fluid, they start moving from blood to the dialyzing fluid. Not only nitrogenous base, excess water, excess salts also can move, can be filtered. And once this blood which is there in the cellophane tube is filtered, now it is passing through the dialyzing unit, gets into the cellophane tube and finally drained back into the human blood through the vein. But before the blood is given back to the vein, anti-heparin should be added. So, this process can be performed for about 2 to 6 hours. That causes the filtration of the whole blood. And this dialyzing fluid periodically it should be removed and new dialyzing fluid have to be added. Otherwise, after some time whatever the dialyzing fluid is there here, because of movement of nitrogenous base, the concentration of fluid and blood may become equal. So, to prevent that one, fresh dialyzing fluid should be added again and again. And this is a temporary relief for the uremia patients, and it is a great boon for the patients suffering with the uremia. But ultimate correction for the renal failure is a kidney transplant, that I will discuss later. Next, the next disorder is glomerulonephritis. Glomerulonephritis is nothing but inflammation of glomerulus that is caused due to the infectious agents present in the blood. The next one is kidney transplantation. Before going for that, renal caliculi. Renal caliculi are commonly known as kidney stones. Kidney stones means not only kidney stones, in the kidney, in the ureter, in the bladder, all these stones are commonly named as kidney stones. What are they? Kidney stones are nothing but they are insoluble mass of crystals of calcium oxalates, calcium phosphates. They are insoluble and their deposition can form stone like structures. They are said to be kidney stones. For kidney stones, the kidney stones can be, the treatment is they can be broken down by using shock wave treatment like lithotripsy, and they can be removed by using the minor surgery also. And finally, kidney transplantation. The ultimate remedy for the renal failure is kidney transplantation. For a renal failure, transplantation of kidney is required, but for that tissue matching and blood matching is required. Though tissue and matching and blood matching is there, still there is rejection. TC cells, especially T lymphocytes, they are involved in the rejection of transplanted organ. To suppress that rejection, immunosuppressor should be injected to the person throughout life to prevent that graft rejection, to protect that transplanted kidney. So, this is regarding kidney transplantation. So, with this I have completed the excretory products and their elimination chapter. In detailed description I have given. Thank you.